So, we are talking of the lymphatic system, and we know that there are capillaries and under the fluid in the capillaries, blood under pressure, it oozes out, and it contains a lot of no RBCs, no large proteins, but the fluid part of the plasma flows out. Along with that goes the glucose, the amino acids and all the small molecules, and they continuously come out, and it is a huge amount. It is almost calculated that as much as 20 liters of fluid may flow out via all the capillaries over a period of 24 hours. That is a lot. Okay. But thanks to the that hypothesis, what was that hypothesis? Very good. What was that hypothesis? Say that again, please. Say that again, please. Say that again, please. Say that again, please. Loudly. Everybody shut up. Again, please. No, he has to say. Others have to. Starling. What is it? Starling hypothesis. Okay, because of Starling hypothesis, that fluid comes out and it again goes back. Okay, and there is but all of it doesn't go back. Okay, and about 2 or 3 liters of it stays there. Nothing stays there. Okay, it keeps on moving, and it's very important because there may be some large protein molecules. We see no system is perfect. Okay, so there are some large protein molecules which have leaked out or oozed out, and then you need to return them to again back to the blood. You have borrowed from the blood, you need to return to the blood, and therefore we have what you call as the lymphatic system. So actually what comes out of the capillaries, it's interstitial fluid. What is it? It is interstitial fluid, it is extracellular fluid. Okay, it's very poor in protein molecules as compared to blood. No RBCs, but short of that it is watery, okay, and it has all the small molecules and all the ions. They are as much as they are in the blood, they are in the same concentration. They are also in the extracellular fluid. Now this extracellular fluid is going to enter into a system of tubes which I will call as lymphatics. Okay, and it's a very tiny tube. It's again made up of endothelial cells, and it is the system of tubes that is going to return the interstitial fluid back to the blood or back to the heart. It is interstitial fluid as long as it has not entered into the tubes. Once it has entered into the tubes, I will not call it an interstitial fluid anymore, I will call it as a lymph. But it is essentially the same thing. Okay, so we have seen this, we have seen this, we have seen this. So we have the lymphatic system, and we have the terminal branches, and the terminal branches get together, and en route they encounter several nodes about which we will focus on in the immunology part, and then finally it will pour. So what is the most important question that you ask yourself is, this is a system that is not driven by heart. There is no heart. Okay. And there is every day over a period of 24 hours about 2 to 3 liters of fluid is coming out, and then it has to flow, it has to go back. Who drives the fluid? Getting the question? There is no heart. Who drives the fluid? The answer to that question is very interesting. Number one, the fluid that is continuously coming out. Where? From the capillaries. Okay, which means it is gently generating a pressure. Okay, which is the fluid is coming. Okay, the fluid has to go somewhere. Okay, as the fluid goes, it generally enters into what you call as these. Is the picture very clear to you? Can you see the red vessel? That is an artery, then it branches, and then the capillaries, and the capillaries run, and then the blue ones which is oxygenated, deoxygenated, and here we have a system of blind terminals, and we'll call this is made up of this is the beginning of the lymphatic system, which is made up of extremely flat and curved like capillaries. They are capillaries. We will call them as lymphatic capillaries, and they of course one flats another, and they make a certain tube, and it is blind at the other end, and in between the two cells, okay, there is a lot of gap. Okay, and one interesting point that you have here is there is no basement membrane. Normally, okay, in all blood vessels everywhere you have, in the capillaries you may or may not have, depending on what kind of capillary you are, okay, that we have done. So essentially on the terminals there is no basement, so just a single layer of cells, and there is a space between the two, and as a result of that the fluid which is being pushed out of the capillaries can gently flow into this system of tubes. Now here the interesting point is, it is something, just imagine there is one cell here, hello, one endothelial cell here, another endothelial cell here, there is a slight overlap, can you see that? And there is a slight space in that, and through that space the fluid trickles in gently. As it trickles in, the volume in this space, the volume in this lumen, what is the lumen? Lumen of the terminal tube of the lymphatic, the lumen here. In that lumen, the fluid is arriving. Are you okay so far? As a result of that, these three cells, they slightly stretch. And the beauty of the cells is that they contain actin myosin filaments. As a result of that, if you stretch them, they will contract. And once the moment they contract, you know, this space between the two acts as a wall, just see, and it closes. Are you with me? So whereas the fluid which is coming under gentle pressure from the capillaries can get into that terminal lymphatic vessel, it cannot go out. Okay, and then the fluid flows, and then as if we were to design, we'll of course put valves there. Okay, just to make sure that the fluid doesn't go back. So we have a system of valves. Okay, I just put this histological picture to give a real perspective of what we have. A muscle tissue. In the muscle tissue, can you appreciate the muscle there? There are all muscle bundles there. Okay, and then can you see that big artery there, branch of arteriole there? But I want to draw your attention to this. The author has labeled as L. What does L stand for? It's a lymphatic vessel. Extremely thin cells are there which are lining, and they they have no basement membrane. They don't essentially form a particular a specific circular. No, they can take any shape depending on the tissue around them. So here we have a terminal lymphatic. There is a cell here. There is a cell here. There is a gap here. The fluid will flow this way it will go here, and once as the fluid accommodates, this is going to swell. Once it swells, it is going to constrict. Once it is going to contract, this flap of the endothelial cell is going to serve as a valve and close, and will not allow the fluid to go back by the same route by which it came. Are you with me? And then it will start flowing, and then beyond a particular limit, then can you see a valve? This actual histological section of a lymphatic. Can you actually see the valve? So the fluid can go here, but the fluid cannot go back because, okay, eventually what happens is, so there is a lymphatic terminal there. The fluids can enter and the fluids can go. There is a valve here. Eventually, as the more and more lymphatic vessels coalesce, which means come together, okay, and they become more in diameter, eventually the larger or the collecting lymphatic vessels we find that they are equipped with smooth muscles. What are they equipped with? Smooth muscles. Have we done smooth muscles in this class? No, we'll talk about it when we do the digestive system, but they are the smooth muscles. But we talked about them 100 times. Smooth muscles, they are the smooth muscles. And these smooth muscles, they show peristalsis. Hello? You get the meaning of the word? We have done it. Peristalsis. What is peristalsis? We have done it with reference to the transport of food along our alimentary canal. Okay, this peristalsis, peristaltic movement. Okay, a wave of contraction that goes along, and there is a peristaltic movement, and the peristaltic movement drives the fluid towards the heart. The lymphatics, it goes towards the heart. Can the fluid go back? No, it can't go back. As a result of the peristaltic movement, it actually creates a negative pressure here. Are you with me? So you are, as you go ahead and as the tubes become larger and larger, and as they get thicker and thicker, covering of the smooth muscles, and as those smooth muscles show a peristaltic movement, and as the fluid is being driven in a particular direction, on the other direction there is negative pressure. So the fluid is being drawn, which is the fluid that is coming out of the capillaries, get into the interstitial fluid, get into those terminals. We'll call it as a lymphatic, and then it will flow along, and it will flow along, it will go towards here. So that negative pressure is very, very important. That negative pressure is important because, you know, some book actually compare it to the terminal serves as a bit of vacuum cleaner. Are you with me? What does it suck? Lymph. What does it suck? It sucks lymphs. Okay, it's continuously lymphatic fluid. It is sucking interstitial fluid and calling it as lymphatic, and that is very important. I'll talk about it again when I come back to respiratory system. That is very important with reference to our lungs. Okay, we have already seen that there are capillaries, and the capillaries in our lungs are under what kind of pressure, please? Hello? Pressure, the blood vessel. Okay, the blood vessel that is going to the lungs is under what systolic pressure? What systolic pressure? Well,
50, 55.
Okay, you can keep on guessing. Okay, 15 mmHg is the average one. So okay, 25.8. I am talking about what? The pulmonary systolic. Okay, I'll go back to systemic. How much? 120, 120, 80, 120. Am I right there? Hello, everybody. Hello, how much is it? Systemic aorta, how much it is? 120, 80. Now I'll take my pressure sensor and put it on the pulmonary artery. Okay, and then pulmonary artery, which is taking deoxygenated blood to the lungs, is showing me what variation? 25.8. You should never make a mistake. 25.8. Average is about 15, 16, whatever. Okay. Now even under that pressure, if the capillaries start leaking the same fluid, do they leak? Yeah, they do. Because the capillaries are there in the lungs. Do they leak? They leak. Now, if that fluid leaks out in the lungs, and if that fluid forms a tiny layer in the alveoli, what are you doing? You are drowning. Okay, you are creating a barrier between air on one side and alveolar epithelium on the other. How is it going to impede with your gaseous exchange? Therefore, we have a lymphatic system that effectively keeps on serving like a vacuum pump and keep on sucking out that liquid, okay, and I'll use the word, keep making sure that the inner surface of the alveoli is relatively dry. Is it dry? No, there is a thin layer of liquid has always got to be there, but make sure that the thin layer of liquid actually remains thin and it doesn't become. The second thing is, this is one point. The second point is, you see why nature is amazing is because every time, you remember when we talked about your calf muscles, two calf muscles, okay, and they play a role in driving the venous blood towards the heart against the gravity. You know, what are we doing? You are using our skeletal muscle to serve a particular purpose. Your entire skeletal nervous system, okay. So whenever you are doing any particular activity, the whole engineering design of your lymphatic system is such that it keeps on pressing these tubes so that they will keep on driving the fluid in one particular direction. And it is so genius. There is nothing else. Every time you breathe in and every time you breathe out, okay, there is a mechanical movement of your thoracic cavity. Am I right? Yes or no? Even with that, the lymphatics are being pressed and released, pressed and released. The moment you press them, the lymph will go towards the heart. Are you with me? So it is so our biology has linked the movement of the lymph in the large lymphatic vessels with our respiratory movement. Next point. We continuously have pulse. So there are blood vessels which are, depending on systole, diastole, they are moving. They are generally designed such a way that they are close to the lymphatic, and even under that pulse pressure, the lymphatic vessel is, and it keeps on pushing so it's the nature, whatever is possible in that scenario, the nature has used it to drive the lymph from because there is no designated heart for that purpose. Okay, look at this. This is the interstitial fluid pressure. Okay. And interstitial fluid pressure means pressure of the fluid has come out of the capillary, it has not entered into the lymphatics, okay, and we are monitoring the pressure. So the pressure may be -2, 0, it's mmHg. You see, this blood has come out of capillaries. We already know that by the time the blood is in the capillary, it may be how much? The blood as it enters into the capillaries about how much? 30 mmHg. Very good, 30 mmHg. Maybe about 8 or 10. Then it comes out. As the pressure goes on increasing, the lymph flow goes on increasing. Means what? The lymph is coming. The interstitial fluid is coming out from the capillary. If you increase the pressure, the lymph flow goes up. Means what? The lymph flow depends on that. Okay, so and there is a direct correlation within a certain limit. Okay. Who can tell me what is the meaning author wants to give us in this slide? Can you make a sense there? There is edema. There is edema means the lymph has to flow, and if for some reason if the lymph doesn't flow, okay, then this may give rise to what? Stagnation of a lymph in a particular area of the body, and then it might give rise to what you call as the edema is of, I may even furnish you with a little more info. Edema is generally considered into two types. One is called as a pitting type and non-pitting type. What did I say?
Pitting type and
not in the pitting type of edema, if if your foot is swollen, you press there and remove your finger, the pit is still there. Okay, what do we call it as? Pitting type. There is other type in which you press there, you remove your, the pit is no more there, the tissue comes back. Okay, so the pitting and not, this is generally the pitting type of edema that you can have in front of you, and then you can have, and then finally you have the, it pours into what you call as the thoracic duct, and the thoracic duct goes into the vena cava, and then it is finally returned to the, okay, I think I'll complete this. Another 5 minutes we'll complete this interesting topic on lymph. Well, I think you can guess what is the image on the left there. We are talking about histological picture through the intestine. Hello, intestine. The moment I talk of intestine, you know that I am talking absorption of food. I am also, you are histological, you know that it is thrown into villi. Okay, and the villi, the purpose of the villi is to increase the surface area. We have done it. Very good. So we are so look at the, so that's the histological picture, and you can those villi, and I am, let us take it one villus, singular villus, and we are trying to diagrammatically show what is in a single villus, and I am sure you can find out there are those red vessels which are the oxygenated, the arteries which are carrying the oxygenated blood, and then it is giving oxygen, and the venous and the blood, and the blue ones are taking the deoxygenated blood away from the villus. But inside that, can you see a little creamish tube there? Okay, that is the terminal lacteal or terminal lymphatic vessel. It's called as central lacteal. What do you call it as? It is called as what? Central lacteal. And if I were to take a transverse section of a villus, okay, it looks something like this. There is a single layer of cells, which is the epithelial cells, which is the inner lining of a villi, or inner lining of the intestine you can call, and the red and the blue ones are of course the capillaries which are bringing the oxygenated blood, giving away oxygen, becoming deoxygenated and going to the venues, etc. And at the center I am drawing your attention to this pinkish circle which author is labeling is what? Central lacteal. What do you call it? Central lacteal or lacteal or whatever. Okay. Now this central lacteal is interesting because, okay, we are talking about the intestine, and it is a digested food, and the digested food again I will divide into 3 categories. I will say that there are small amino acid molecules which are small carbohydrates like glucose and others, they are all small molecules. Now listen to this, whether it is amino acids or glucose or carbohydrate, digested carbohydrate molecules, I will put all of them together. Are you okay so far? Whereas these molecules will be taken up by the blood vascular system, and they will carry to the blood will carry those molecules. So the molecule, the food that you have absorbed, not all the amino acids and the carbohydrates, glucose, etc. They will be taken to the liver by what you call as hepatic portal system. Now, what is hepatic portal system? Hang on, I'll talk about it when we talk about the digestive system. But the only point that I want to make is that the blood is being taken, all those nutritious material, not all amino acids and carbohydrates are being taken from the intestine, taken where? Taken where? How? Just remember the name. What blood vessel? Hepatic portal system. Okay. But the digested fats, listen to this, the digested fats are poured into the lacteals, and the digested fats are absorbed and taken, okay, along with the lymphatic system to the vena cava, and then to the heart, and then to the general circulation. So whereas the carbohydrates, digested carbohydrates and digested proteins in the amino acids are essentially taken where? From the digestion first to the liver for further processing. The lipids shunt it, okay, they don't go to the liver, they go to the general circulation elsewhere also. So please appreciate this very interesting role of the lacteals with reference to the absorption of fats. Okay, I am sure this is a node. I just put the slide here wherein the lacteals are flowing from all over, and here the of course you know the T cells and the B cells and the immune function will take. Yeah, please.
So could you go to that villus? So how is the distinction? How are glucose and amino acids segregated from fats and then?
Okay, I didn't want to talk about it, but okay. It is all dependent on the size. The fat molecules are organized into larger particles which are called as chole-microns, chole-microns, okay, and they are very large ones relative to the size of amino acids are so whereas the smaller ones can enter into the capillary, the larger ones like chylomicrons, which are containing different kinds of fatty acids and all the digested product of the fats, okay, they are organized, okay, they are not traveling as a single independent fat molecule. No, they are organized as certain very large globules, and those ones which we call as chylomicrons, they cannot enter into the capillaries. They are big enough, so they have no that they enter into the lacteals, central lacteals, and then they are carried. It is they are differentiated on the basis of the size.
So they can trickle between.
Oh yeah. The lacteals, the endothelial cells of the lacteals of far larger space. Far larger space, these fat molecules. Fat molecule is the wrong word I am using. The chylomicrons, okay, they can enter, but not but you got the answer, right? Got this sense, okay. And this is the last slide. What does this slide tell you? You are talking of a disease. What disease are we talking about? Why is the disease? What is the root cause, etiology of the disease? There is a nematode. Okay, there is a nematode, Vicoria bancrofti. Okay, that is the name of the worm. Okay, and that worm tiny there. Can you see that worm? It's a nematode. It's a nematode. The vector for that is a mosquito again. So mosquito does 10 bad things. One among these is those worms. Okay, filaria worms. Okay, and those filaria worms, if they get into your body, they specifically sit into the lymphatic network. Okay. And then over a period, nothing happens right away, but over a period of time, as the flow of the lymphatics or lymphatic fluid is obstructed, then the tissue responds, and the tissue responds by generating what you call as there is a formation of fibrous tissue, and as a result of formation of fibrosis, you have this elephantiasis or of this rather ugly looking disease.